It is our most advanced technology and was mature at the time when we all were hit by the pandemic, to take it out of the toolbox and to use it as our vaccine technology. And for mRNA, it was the baptism by fire.
That was Dr. Özlem Türeci, the Chief Medical Officer of BioNTech. In early July, science journalist Kai Kupferschmidt spoke with Dr. Türeci and Dr. Ugur, the Chief Executive Officer of BioNTech. They co-founded the company in 2008. Dr. Sahin and Dr. Türeci were elected EMBO members in 2021. Kai spoke with them about their journey from physicians caring for cancer patients to physician scientists to entrepreneurs, and how BioNTech, a company with a strong focus on cancer, managed to develop the first licensed COVID-19 vaccine in less than 1 year. Welcome to the EMBO podcast.
Thank you so much for taking the time. It's a pleasure to have a chance to talk to you. And even as a science journalist, it doesn't happen that often that you talk to people who actually kind of had this immediate impact on your life. I have my 2 shots of BioNTech. So I guess this must happen a lot to you now, that people kind of, go up to you and just tell you, thanks.
Yes, yes, they do. I think for all of us, and we experience this in the same way, getting the vaccine is some sort of a relief. And so going back to freedom, and people want to share this relief. It's normal.
I'm curious, you're probably 2 of the earliest people who got the vaccine, right? Are you kind of following your own antibody titer a little bit along and kind of to get a sense of how much it wanes?
No, we are not doing that. We were not among the first ones. As you know, the first ones were the most vulnerable ones, so we waited until it was our time to get the vaccine. And we are not monitoring our antibody titers. We are relying on the data which comes from the bigger studies and on what we find out in terms of scientific insights when boosters would be required.
Can you take me back to the beginning of 2020? I remember that time very well. Ugur, I read that you read, I think, an article in The Lancet in early January about the cases in China. What exactly went through your mind?
Yes, so I read a publication in Lancet describing, it was the first scientific publication describing cases in Wuhan, and the publication showed that this new outbreak has the full pattern of a pandemic threat, and the most important new information in this publication was that there were individuals who did not have symptoms, no fever, even though they were positive for the virus. Yeah? And at that time, there was no restriction for traveling for people who did not have symptoms. And so it was very clear statistically, I did some calculations, it was very clear that this outbreak already is in a pandemic phase. And then, of course, the next type of thoughts were, what can we do? Yeah? And is our technology suitable to develop a vaccine? And understanding that we had a chance to make a difference resulted in a more or less obligation to start the project without asking.
Yeah. Sorry to interrupt. Let me just stick with this for a second, because I'm really struck that you say, I mean, this was January when you had this realization. And I remember very vividly in the middle of February, when we had the cases already in Italy and Iran, and trying to convince people that this was, that this was already out of control, basically. What do you think you saw that other people missed? Because there were quite a few infectious disease specialists at the time still, even when the public health emergency of international concern was declared by WHO, I think 30th of January, there were still a lot of people saying, this is wrong. So what do you think put you in a position to make that call, the right call in that case?
Yeah, so first of all, I think many people, not only I and our team, but many people saw that because the mathematics was very clear. Yeah, but what limits us as human beings is the past experience, because this did not happen the last 50 years in this way. Yeah, people continue to do their normal business. Yeah, and as a scientist, you have to disconnect yourself from the past experience. It's not about experience. It is a new situation with a new pathogen, which fulfills the full pattern of an outbreak which can't be controlled because of the combination of high infectivity and the more or less immune naive human population and the high transmission rate and the pathology, and because asymptomatic patients could act as a global carrier, yeah, to spread this disease, and it was just too late. And then it's like it's a full pattern. There is the evidence, the mathematical evidence is there, and it's just only a matter of time that other people will realize that. And we started before without losing time.
Right. Yeah, and I'm always struck that humans, we're just terrible with exponential growth, of course.
Yeah.
So what were the kinds of decisions that you made then at the company? What changed for you in January?
We deployed teams to work immediately on the work streams which are needed for drug development. So basically at that weekend when Ugur, we realized that this would happen and we had to act, there was already a list of constructs, vaccine constructs, which went into which were then initiated. The cloning was initiated. So cloning was one, preparing the experiments for preclinically, comparing the different constructs, starting to design a clinical trial. Shortly after that, first contacts with regulatory authorities, with the Paul-Ehrlich Institute, to start a dialogue early on, thoughts about manufacturing and whether we would have the capacities. So everything was started and put on tracks and then escalated and accelerated over time with more information on the reality of the pandemic coming in.
So you started very, very early compared to others, and then the other thing that you did was to partner up with one of the big players that you mentioned earlier, in this case Pfizer, who you already had a kind of a business relationship with. What made you take that decision?
It was very clear that we had the skills to design a vaccine and to engineer and understand immune responses, and we could also do the phase 1 clinical trial. But we did not have the skills to do the global phase 3 clinical trial, which is required for efficacy testing. Therefore, we decided to identify a partner, a pharma partner who can do that. And since we had already a collaboration with Pfizer, and since this collaboration was really an excellent collaboration with also Pfizer, dedicated on a scientific approach driven by data, we asked the colleagues from Pfizer whether they would like to join us.
It's such an important decision in a way, and I want to understand a little bit how you think about this, because a lot of the commentary that I've read, or a lot of what I've seen about mRNA, it talks about the BioNTech vaccine or the Moderna vaccine as it's all about the technology. This proves mRNA is great. If you look at what actually happened, you could argue the Novavax vaccine, for instance, is probably at least as good. It maybe has a little bit milder side effects and has a similar efficacy. it just came a lot later. And one reason that it came later, I mean, they applied, I think, at the same time for the clinical trial, but Novavax didn't partner with a large company. And by the time they got started, the cases were lower and all of these things. So when you look back at this story, how much of it is really about this mRNA technology? Because we now know that a lot of different technologies are able to produce decent vaccines against this particular virus, and luckily for us. But how much of it is that, and how much is you getting started very early, making a very smart decision about who to partner with, and then just the clinical trial being run really well and fast?
So a number of things, as always, there is not one answer. A number of things have to play together, and therefore it's very important to map those threads. You have to weave together very early on and take care of them. And one of those was this partnership. So mRNA technology, without now comparing it with others, has a couple of features which plays towards being able to be fast, being able to be agile, and have some versatility. It's a synthetic molecule which can be easily produced, which can be relatively easily upscaled, and which can be also relatively easily technology transferred when it comes, for example, to cooperating with partners. This all certainly takes time and so on, because GMP manufacturing and GCP clinical trials have their regulations with which we have to comply. So the technology is one thing that it allows to be fast. Another thing is that this technology was really mature. We had not only developed it over years, we had already translated it. So there were clinical trials going on all over the world, hundreds of patients already treated with our cancer vaccine, manufacturing capacities at a stage where we alone had already the technology, the clinical trial material technology in place, and were able to deliver material for the entire clinical development. So this all was in place, but it was also clear this expansion, the worldwide clinical trials, the worldwide connections to regulatory authorities, and the further expansion of manufacturing capacities need additional support. And this is the case for many of the technologies, and we reacted early and put this in place with our partners.
In your construct, you use the two proline residues that Barney Graham kind of, in the pre-fusion spike found to be, to stabilize that conformation. And as somebody who, moved into this whole space, kind of ad hoc when the pandemic hit, I'm just curious how you make these decisions. Did you just read the papers and immediately realize, okay, this could help us, or how do you go about that?
Exactly. The weekend after reading the Lancet paper, we read about 100 publications, and the data that Barney had described for SARS, as well as the MERS vaccine, plus the crystal structure that was published, convinced us that this is a good way to go.
Okay, and the other thing, because we keep talking about the mRNA and we talk much less about the packaging of it, right? The nanoparticle technology. We don't have a lot of time, but over the years, what's kind of the main theme that has emerged for you? What works? What have been the improvements there? We talked with the mRNA about the pseudouridine, for instance, but with the nanoparticles, what were the major steps there?
Yeah, it is like in the messenger RNA. There is not one single innovation. Yeah, so you have really to combine many innovations and improvements. And for the nanoparticle, it is important to ensure protection of the mRNA, which is accomplished by many nanoparticles. On the other side, ensure a high transfection rate. Yeah, and we had tested hundreds of formulations and discovered this formulation, which is from another partner, from Acuitas, to be one of the best, fulfilling the mode of action in which we were interested, a delivery mechanism that allowed us to bring the mRNA to lymph nodes where immune responses are primed. Yeah? And therefore we decided to go with this empirically identified formulation as the one to be used in combination with our mRNA.
So and we ended up with the BioNTech vaccine, with the first person being vaccinated at the end of 2020 already in regular vaccination after approval. And I'm curious how you think about the fact that we have this amazing tool and it has saved countless lives, but of course right now we're in a situation where it could also have saved many more lives. But we're now in a situation where we're vaccinating teenagers, in some countries like Israel or in Germany increasingly, the US, and then there's other places in the world where there's no vaccine even for frontline healthcare workers. You're scientists, you're also entrepreneurs. There's so much involved here, but what is the role of you as the scientists who developed this in this? How do you think about this? And is there where's your responsibility in a sense? Where does it end? What can you actually do?
The question is equality of distribution. You are getting to, I guess, Kai, and that has been of importance for us from the very beginning. So we repeated the mantra others have designed. No one is safe if not everyone is safe, and we meant that. And from the very beginning, I mentioned those work streams. We also initiated work streams which would allow, that takes time, and therefore we are not yet there, which would allow also distribution in terms of storage and transport conditions into low and middle income countries. So we prepared for that, and as you might have also recognized, we overperformed in terms of what we promised to deliver. We started with 1.6 billion. We are at 3 billion because we managed to beat our own goals. And in fact, it is not as bad as sometimes described. We will via COVAX deliver 40 million doses to low and middle income countries, and we have committed to 2 billion doses within the next 1.5 years to low and middle income countries. The first 1 billion within this year, which is 40% of our production capacity.
Yeah, may I add? So besides really delivering the vaccines to these low-income countries, this is the one thing that we are committed to do. We believe it is important to enable that, for example, in Africa, dedicated regions have access to the technology. So we are committed within the next years to identify partners with whom we can transfer our technology, build manufacturing capacity in Africa to ensure the next time when such a situation happened that these low-income countries have everything in place to manufacture their own vaccines.
Before we run out of time, I want to get back to the original, your original mission to oncology. And you've had, this very fast, amazing success with this vaccine against COVID-19, and that, of course, stands in stark contrast to how hard it has been to develop anything in the cancer field. How do you see, the future? Is there anything you can take from this experience as well with COVID-19 that will help you with your original mission?
Yes, definitely. So it was using our experience in cancer vaccines to cross-fertilize the COVID-19 development, and now this is given back to cancer, what we have learned there. And it's not only what we have learned, Kai, it's also about, for example, regulators learning more about the technology and about safety profiles and also about learning how to upscale manufacturing and refine that, and learning about the immune response patterns. So this all will definitely be tailwind for us, for our developments in the cancer realm, and how we will modify and improve and accelerate our efforts to have cancer vaccines.
Right, and obviously you're also working on other infectious diseases. I sometimes feel like people are, I get a lot of questions about like, oh, is this the technology that's going to give us an HIV vaccine? And my personal take is always the problems that we have in developing an HIV vaccine have very little to do with the technology we use to deliver the vaccine, and a lot to do with the pathogen itself. Where do you see in the infectious disease field?
Yeah, so the what drives us is really medical need, and there are a few pathogens for which there is a huge medical need. For example, tuberculosis. So we believe that tuberculosis, that mRNA could be very suitable for development of a tuberculosis vaccine. So we started also in partnership with the Gates Foundation already almost 1.5 years ago, a project to develop tuberculosis vaccines. So we plan to bring such vaccine candidates most likely in 2022 into clinical testing. The other great challenge in the field is malaria.
I read, I think it was in the profile in the Financial Times, that when you got married, you went back to the lab afterwards on that day. So clearly you're both very driven. What drives your interest in science and health and being a doctor? Where did it come from? Where did it start for you?
Yeah, maybe I start. So for me, it's two reasons. One reason is really curiosity, and I love science. I love mathematics. I love to understand things, try to find patterns, and try to understand. So that's one motivation. The second motivation is really to be able to help and to make a difference for individual lives, and try to solve medical problems, by identifying the ground challenges and addressing them. It's the combination of two things.
Yes, it's pretty similar for me. My dad was a physician, and I grew up experiencing what it means to treat patients and what fulfillment it in particular means. And later, when I was at medical school, I did my doctoral thesis in molecular biology and deep dived into the realm of science. And this divergence, or this gap, on the one hand seeing that as a physician I could not offer much to my cancer patients, and on the other hand seeing the entire potential we would have if we would translate science and technology into treatments immediately. That was something which motivated me, together with Ugur, to select the special path of career. And it also involved becoming an entrepreneur out of desperation, because this seemed to be the only way to bridge this gap.
Do you remember any specific moment or any experience when that became clear to you? Because in my experience, for a lot of scientists, it's still, I mean, that is a very big switch to go from being, a curiosity-driven scientist to trying to get an application out there and as an entrepreneur, with all the hurdles that entails.
It was not just a moment. It was really an understanding what is possible in a purely academic environment, and what needs to be done to really bring new type of treatments to patients, and not only to the few patients within early clinical trial, but really develop new medicines which could change the standard. So we did our research in the university hospitals and came up with new concepts and tried to collaborate with industrial partners, to convince them to take these concepts, to use our targets for development of new treatments. And working with the partners, we realized that after a few years, even though the cooperation went well, the pharma partners decided to stop because the leadership changed or because of structural changes. And we realized it is just not possible to do it by collaboration. And it was also not possible to finance that, to finance clinical trials, to finance GMP manufacturing of our compounds. So the only way to go was to start a company and try to get the financing to do all these things.
So what was the most important thing maybe that you learned with that first company that wasn't in the books? What would you have hoped you would have known before maybe?
So for me, the most important aspect was really to take full responsibility. Because the idea, even when we founded the company, the idea was we ensure as founders that we get the financing, and then we hire company people who do the research and with industrial background. And we realized after a few months this is not going to work. We have to really deep dive into the matter to avoid that this is going to be a failure. So that's the very first thing that I had learned. And that if you start a company, if you start a new institution, you have to be responsible.
Yes, I cannot agree more. It's really this understanding that you need skin in the game yourself. And I fully believe that the new breed of biotech or biopharma executives are scientists, because science really has to lead the way. The innovation cycles also for healthcare medicines, for healthcare products and medicines, will be absolutely science-based and faster. So I agree to Ugur, that's an important learning.
I mean, one of the interesting things to me is that Ganymed, of course, you started that actually trying to develop antibody drugs, right? Can you tell me a little bit about that? Because that's quite a long way away, of course, from the mRNA that everybody now kind of knows you for, I guess. At the time, what was promising to you about that? And maybe you can also update just briefly, because I realized the company was sold, but I think there's a phase 3 trial going on, right, of the compound.
So at the time we founded Ganymed, we had a very active and busy research team in an academic team at the University Hospital in Science, and we were pursuing numerous different immune therapy approaches, including mRNA antibodies and other formats, because we were interested really not in a specialized way on one type of immune therapy, but in immune therapy combinations. And the antibodies we were developing were ripe to be spun off into a company. That was the reason why Ganymed was very focused on that type of personalized approach, namely having a target which is very precise and specific, and developing antibodies which are a well-known established technology, easier to industrialize against this target. And all the other activities remained in the academic space, and we continued them. And so that was the reason why Ganymed was so specialized and focused on antibodies. And later on, when we came to a point where we in fact showed in randomized phase 2 trials that the antibody works, we were also aware of the fact that we would not be able to finance and also conduct operationally as a small company phase 3 trials. And the usual business model for biotech companies founded by scientists is and was also at that time to then sell to pharma, which has the machinery to do this. And the pharma partner who acquired the technology, Astellas, is running now 2 big phase 3 trials, global trials, and additional phase 2 trials in other indications. That is still going on.
I find it interesting that the sale of Ganymed was one of the biggest deals in German biotech at the time, and I guess it still is. And yet, it feels like that's something that in Germany I didn't know about it, to be honest, and I haven't seen it celebrated in the way that maybe it would be in the US. And I wonder how you feel, academics kind of think about this. Like, how did they react to this kind of news? How do you feel this is perceived in academia more generally?
Yeah, so first of all, what was great with the experience of our development, antibody development at Ganymed, was that the company was almost not known, even not for experts. And we just did our research. We started the phase 2 clinical trial, and this trial was one of thousands oncology trials running worldwide, and no one really noticed. But at that time point, when we got the results, we just sent an abstract to ASCO, which is the greatest cancer conference. And this abstract was selected as one of the best presentations for presenting to the press. So it was really interesting that we just included the findings of the trial, which were very convincing, and the scientific approach. The recognition happened. So within a few days, the company became famous, and we had a lot of interest from pharma, and it took about 8 to 10 weeks later that the company was sold to Astellas. So that means you don't need to make a lot of noise, to be recognized. It's just having the data, the science, and if the science and the data confirms, then you will be recognized. And this is a wonderful experience, and we do not seek recognition for what we do. We just want to develop our medicines, and if they are successful, we want to bring them to the patients. Yeah.
So clearly, there must have been something there that triggered you enough to want to found another company, because you then founded BioNTech. What was the idea behind that company when you started it?
So our original motivation as physicians is to help individual patients. And when we started our personalized cancer immunotherapy project with the antibody, we realized that 70% of patients had to be excluded because the target was not expressed. And that's not the way which is satisfying for a physician, because a physician really wants to provide the best possible treatment to the patient. So we were from the very beginning interested in individualized medicine. The idea of individualized medicine is to analyze the targets which are expressed in the tumor of the patient, and develop a treatment which is really tailored to the antigens in the individual tumor. And that's what motivated us also to start our mRNA research. And BioNTech was founded, it stands for Biopharmaceutical New Technologies, was founded to come up with a number of technologies that would allow this personalized tailoring to enable treatment of individual patients. So that was the motivation behind the initiation of BioNTech.
So at what point did mRNA become really the focus in a way? Because, I mean, like you said, new technologies is the NT, so in the beginning there were different approaches that you were looking at.
Yeah, so I would not say that mRNA is the focus, because we are solution and goal driven, and the goal is clear. We want to provide better medicines, and mRNA has been one of our technologies which we started in the early 2000s to develop and to adapt for different therapeutic modalities for vaccines, but also for expressing antibodies or cytokines. And it is our most advanced technology and was mature at the time when we all were hit by the pandemic, to take it out of the toolbox and to use it as our vaccine technology. And for mRNA, it was the baptism by fire, and we could show that it works, but we have other technologies in which we also believe in our toolbox, which we think can also work for other purposes.
So there's this idea that, you know, in the beginning, it seemed full of promise. I mean, it's something we see a lot in biotech, right? You have this, in the beginning, someone has a nice idea on a blackboard, and it's like, wow, if this works, it could do all these things. And then you hit the first snags. In this case, it was very immunogenic, the uridine especially. And so you ended up, basically, there wasn't a lot of funding and people didn't really develop it further. And you just have to look at the history of Katalin Karikó at UPenn, right? And so at what point do you, how do you look at this technology and then say, oh, but I think, you know, this is now ready, or we can use this? What got you excited about it in a way, and when?
Yeah, it was in the early 2000s, and this was pre-BioNTech. This was in our academic group where we were looking into different technologies to serve as a vaccine, meaning to deliver the information of an antigen to the organism in order to induce immune responses. And we tested all the different technologies, including those which were hip at that time, and then mRNA was included in this. And at that time, mRNA was overlooked because it was not potent enough, specifically because once mRNA was brought into a cell, the half-life of the RNA and the translation efficiency to protein were so low that the amounts of protein you would get would not be sufficient. And we, however, saw that on the one hand, you want something which is transient due to safety reasons. You don't want something to integrate. So there was a potential, or there was a feature which was interesting. On the other hand, we were sure that we would be able, with sufficient time and effort invested, to tweak the pharmacokinetics of the mRNA. And third, mRNA, uridine-containing mRNA, has intrinsic adjuvanticity. So also that appealed to us, that we had this immune modulatory effect. And we invested several years, throughput screening technologies and so on, to modify the non-coding regions of the mRNA, the cap region, the UTRs, the poly-R tail, how to purify it, in order to ensure that we get this pharmacokinetics and the translational efficiency right. And in fact, what we achieved by combining better designs of those elements was a more than 1,000-fold increase of protein yield in, and this is now important, in human dendritic cells, because this is cell type specific, and we were immunologists, so we knew which cells we want to target. So that was one of many buckets of improvements we developed over time. And another improvement is the nucleoside modification, which was developed by Kati and by Drew, which makes mRNA, which blunts the immune modulatory mRNA feature, which is one of the modifications which on the one hand ensures that mRNA is translated longer, so it also plays into the translation efficiency part, and also blunts the immune modulatory effect. The uridine mRNA, that one we developed into clinical stage. For our cancer vaccines, we have uridine mRNA. We need the immune modulatory capacity because cancer is a much higher hanging fruit as compared to viruses. We also developed the first and I think only intravenous formulation for mRNA, which means a specific lipoplexes which carry the mRNA body-wide into the dendritic cells in lymph nodes in order to get the expression of the antigen in the right place in the body. And sort of this and even more investments into improvements over the years have brought us to the place where we are now.
So you were developing this technology in the beginning, of course, still with your eye on cancer, on oncology, right? And like you just said, in some ways, viruses, of course, are way lower hanging fruit, and you don't have to be personalized for them. I mean, personalized for the virus, I guess. But in what way, at what point did you kind of realize, well, we could also do something in infectious diseases? Was that an easy transition?
So infectious disease vaccines were an obvious application from the very beginning. But the key limitation for development of infectious disease vaccine is not the technology, but it is more the way how infectious disease vaccines are developed. There are only a few players worldwide in the infectious disease vaccine field, and these are large pharmaceutical companies which can do large clinical trials with 30,000, 40,000 subjects and are able to ensure the worldwide supply. And this was not BioNTech, so it was very clear and that infectious disease vaccines is something we could consider later on. Because we were not interested in just competing with existing products. We really wanted to develop products which are not existing. And of course, the situation changed when we realized that this outbreak is going to become a pandemic, and then we were back in this motivation, to develop something which could help humanity.
Obviously, you started as physicians. You're not treating patients anymore, I understand. And I mean, obviously, you're doing a lot of things. You're still teaching. You're running the company. Was that a hard decision? Do you miss it? Like this idea of having to almost be a full-time entrepreneur and not getting to see the patients anymore?
Yes, it was a tough decision for both of us. But seeing patients means really 150% commitment, because it's not technical. It's really a humanitarian and emotional thing to do. And when we decided to quit that realm, we also said if we are now quitting that, we need to make sure that the investment of effort and time into developing medicines is so successful that at the end of the day it pays off for all the patients we could not treat in that time. So it was on the one hand a difficult decision, but one which was a continuous incentive and motivator for us.
Thank you so much for taking so much time. I really, really appreciate it. It's been really interesting.